Okay, so here we go with the second part of Chapter 9. We talked about the general senses in Chapter 9. Now we're going to talk about the special senses. And the special senses are smell, sight, taste, balance, and hearing. So olfaction is smell, vision is vision is sight, obviously. Gustation is taste, equilibrium is balance, and hearing. So we're going to talk about smell and taste very briefly, and we'll spend some time on hearing and equilibrium, which all happens in the ear. And then sight as well. So we'll spend quite a bit of time on hearing and equilibrium and sight, and spend just a little bit of time on smell and taste. The processes there and the mechanisms that happen aren't nearly as intricate as in the eye and the ear, so we won't spend as much time on them. These special sensory receptors are much more complex. Let me get my slideshow going. Than the general senses, and they're located in sensory organs like the eye, the ear, right on the tongue, in the nose. So, and they're protected by the surrounding tissues. Information, so action potential information from these special sensory receptors are located, as the information basically is distributed to areas in the cerebral cortex. And there's different smaller areas of the cerebral cortex they're also called cortexes. Like the visual cortex is basically where information about sight is sent from the sensory receptor cells in the eye. There's a olfactory cortex where sensory receptors in the nose for smell send information, and it's integrated there. There's a gustatory cortex. We'll talk about those kind of pathways as we go. So olfaction and taste receptors, we're going to overview some stuff first and then get into some specifics. Olfaction, the sensory receptors are basically modified neurons. Gustation sensory receptors are actually specialized receptors that communicate with a sensory neuron. So sometimes special sensory receptors are modified neurons, or sometimes they're actually their own sensory cells themselves. And olfaction and gustation basically have sensory receptors that are embedded in epithelia and exposed to the external environment. So think about it. When you smell something or when you taste something, you have exposed areas of your nose and in your nasal cavity and your tongue to the external environment. So the sensory receptors are actually located very near that area, but they are protected by some of the epithelial tissue that they're embedded within. And then they integrate information to the central nervous system by way of sensory neurons. So take a look here. The olfactory epithelium are these cells here, and that olfactory receptors, sensory receptors, embedded in that epithelial tissue. Same thing over here with a taste receptor. This is a taste receptor, and there's other cells here that are accessory cells that kind of help protect those. But there's little tiny sensory receptors located here, where like a chemical that you in your food would activate that receptor, and then the cell itself, the sensory cell, the taste receptor, would send a signal. There would be like projections of the afferent neuron. Hearing and equilibrium have special sensory receptors as well, but I'm going to spend just a few seconds on this because we're going to spend a lot of time on these later. There's sensory receptors that are protected from the external environment. So whereas like taste and smell have receptors that are exposed to the external environment, hearing and equilibrium, the sensory receptors for those are embedded in a bone in the skull. They're actually embedded in or protected by the temporal bone in your skull. And there's sensory receptors called hair cells for both hearing and equilibrium that are mechanoreceptors. Remember, mechanoreceptors respond to membrane distortion to activate and generate action potentials. And they send information. They're basically surrounded by supporting cells and monitored by dendrites of sensory neurons. We'll talk about that as we get into those areas. So here's a hair cell, right? So right in here, there's your outer ear, right? There's your ear canal. We'll talk about what that canal is later. And your eardrum. There's a whole nother cavity here until you hit the inner ear. And this inner ear is where the sensory receptors, the hair cells, are located for hearing in this circular structure, and equilibrium in the rest of the structure here. Well, again, we'll come back and talk about what these are. Remember, we talked about the vestibulocochlear nerve, one of the cranial nerves before, that innervates the inner ear. And that vestibulocochlear nerve, where's my cursor? Keep losing it, is right here. right? Here's the vestibular branch and the cochlear branch of the vestibulocochlear nerve. So here's your hair cell, and basically this hair cell has structures on the surface of it called stereocilia, and when they bend in either direction, either stimulates or inhibits this cell from generating an action potential. And that is literally the stimulus. When you hear something, those hair cells are activated and they're generating action potentials and sending them to the sensory neuron here. Olfaction is your sense of smell. Again, we're going to spend just a little bit of time on overview, and then a little bit of time on smell and taste, and then most of our time in this unit on hearing and sight. So olfaction is your sense of smell. What cranial nerve is responsible for your sense of smell? So what cranial nerve is receiving information from the sensory receptors here? It's one of the 12 cranial nerves. So if you can remember that, you're in pretty good shape so far. Right, it's cranial nerve 1. So cranial nerve 1 is your olfactory nerve. So your olfactory nerve has olfactory epithelium associated with it in the nasal cavity where there's olfactory receptors. So in the nasal cavity, there's olfactory organs there that an odorant or a chemical, so a smell, right, stimulates the olfactory receptors. And here's the pathway that it takes to get to the olfactory cortex in the brain, that area of the brain that is the kind of receiving center for olfaction. So sensory neurons are stimulated by a chemical. So the stimulus to activate those sensory receptors, remember we talked about stimuli and receptor specificity. So the receptor has specificity here to chemicals in the air that you would smell. So chemicals here, axons leaving the olfactory epithelium basically make their way through the ethmoid bone. Remember we talked about olfactory foramina, the holes that olfactory neurons penetrate through on a specific part of the ethmoid bone and communicate with the olfactory bulb, the first part of the olfactory neuron, cranial nerve 1. The olfactory bulb has axons that leave the olfactory bulb and you basically utilize the olfactory tract to communicate action potentials from the olfactory bulb all the way back to the olfactory cortex. And the olfactory cortex is mostly where information is received, but it's also received by the limbic system, which is responsible for basically emotional responses to food or to smells. Like if you smell somebody or something, it reminds you of something. So that's your limbic system receiving that information going, oh yeah, I know, that makes me happy or that makes me sad. And the hypothalamus as well. So here's what it looks like, right? Here's olfactory epithelium, right? The olfactory epithelium has embedded in it olfactory neurons, right? And with sensory receptors located on them. So chemicals activate those sensory receptors and they send information to the olfactory bulb and then along the olfactory tract back to the olfactory cortex, which is in the cerebral cortex of the brain. Your sense of taste is gustation, and you have taste receptors located on your tongue, basically the superior surface of your tongue that are next to the pharynx and the larynx, so your throat and your voice box. And you've got these structures, bumps called papillae. They're called lingual papillae, and they're projections on the tongue that are lined by epithelial tissue, specifically stratified squamous epithelial tissue. And embedded in that tissue, you've got taste buds. So there's 1, 2, 3 here, different types of papillae. Taste buds are not the same thing as papillae. Taste buds are located embedded in the epithelial tissue of papillae. And circumvallate and fungiform papillae specifically have taste buds. The cranial nerves that innervate the tongue are the vagus nerve, glossopharyngeal, and facial nerve. So remember, we talked about these when we talked about cranial nerves in the last chapter, in chapter 8. So the glossopharyngeal nerve is basically responsible for about 1/3 of the sensory information coming in from the tongue. And then the facial nerve is responsible for the other 2/3. The vagus nerve has basically information coming in from taste buds on the surface of your epiglottis. And your epiglottis is one of the structures. I got a puppy. Hang on. Okay, back where we were at. So, not from the currents. Yeah, the cranial nerves that innervate the tongue. And again, taste sensations, the four primary are sweet, salty, sour, and bitter. Umami is a fifth that's detected by receptors that are sensitive to like the building blocks of proteins, which are amino acids. Okay, back to it. So basically these umami is detected by basically when there's protein or amino acids broken down, small nucleotides, and basically like the taste of like Parmesan cheese. Think about that. That's an umami type of stimulus. Water receptors are also concentrated in the pharynx, which is your throat, and provide information to your hypothalamus for water balance. So those, again, your hypothalamus has your thirst center. So that's a really important area where the tongue and the throat are able to communicate there about water balance and regulation. So here's this. There's a circumvallate papillae. This is what they look like. So this is their figure. Right there are taste buds. So these little structures here are taste buds. And if you blow that up to a larger structure, right here, there's actually a taste cell. We've seen this before. So here's the epithelium where the taste buds would receive the chemical signal. And then here's a neuron that sends that information about that chemical signal back to the brain, to the gustatory cortex, that area of the brain that receives information about taste. So now we're going to move on to the ear, and we're going to spend the rest of this lecture here, and then we'll have a whole new lecture, which would be 9C for the eye. So we got a lot going on in the ear and the eye. Smell and taste are relatively simple. Ear and the eye are pretty complicated. So we're going to talk about accessory structures of the ear and basically move from the external ear all the way to the internal ear and talk about how a sound wave makes it from the external ear and all the external accessory structures to the middle ear and all the way into the inner ear. Because basically this sound wave gets communicated to your eardrum, and then from your eardrum to the inner ear, basically, is the middle ear. There's another form of communication. It's no longer a sound wave, but there's actually like mechanical events that are happening. And then the inner ear basically takes those mechanical events and turns those mechanical signals into an action potential. So there's a lot of stuff happening here. So follow me along this journey. Basically, the visible portion of your ear is the external ear. So like what you can stick your finger in or a Q-tip in is your external ear. Basically, the job of that is like a satellite dish. It's collecting information, collecting and direct sound waves towards the middle ear. So you've got external, then middle, then inner ear. And the external ear takes those sound waves, collects them, and communicates those sound waves to the middle ear. The middle ear then takes that information and communicates to the inner ear. So we've got a three-step process here, really. And the elastic outer cartilage basically creates the auricle. So what you can grab a hold of, or what you would pierce with an earring, is your auricle. So this outer kind of like satellite dish shaped structure is your auricle. And that collects sound and sends it to the external acoustic meatus. We have learned this structure in the skull. And I said you could stick your finger in your ear. That's the external acoustic meatus. So where you would put a Q-tip internally, where you're not supposed to put a Q-tip actually, is basically that passageway headed into the temporal bone. And it's lined with small hairs and glands called ceruminous glands that secrete this earwax, which is called cerumen. So your earwax, you're not actually supposed to dig that out of there with a Q-tip. It's supposed to be there. Cerumen, this earwax, helps slow microorganism growth, preventing infection. And the cerumen and the hairs there also prevent foreign objects and insects from actually reaching the internal delicate structures. So I'm not going to tell you to stop cleaning your ears out with Q-tips, but I will say that you should stop cleaning the tube with Q-tips. Basically, cerumen is produced and then it gets forced out of the external acoustic meatus towards the auricle. And what you can actually see visibly, you should wipe away with a Q-tip, but don't stick the Q-tip down in. So there's words to live by from Dr. Hill. So here's your external ear visibly. This whole area, this is the auricle, right? And then the external acoustic meatus all the way to the tympanic membrane, which is the eardrum. That designates the division between the external and the middle ear. So your eardrum is the first dividing point, the tympanic membrane. All of this is external ear. And the job of that external ear is to just funnel sound waves to the eardrum or the tympanic membrane. And that tympanic membrane vibrates when sound waves hit it. That's really important for the next couple of steps. The middle ear is actually a cavity. It's called the tympanic cavity. And the one side of that cavity is the tympanic membrane, the eardrum. The opposite side of that membrane, or of that cavity, is called the oval window. And we'll talk about that next. But within that cavity, you've got a tube called the auditory tube or eustachian tube that connects your middle ear to your throat, your pharynx. So if you've ever had, like, been on an airplane and your ears popped when you get up to higher altitudes, there's part of that tube that opens up a little bit and pops, and it helps regulate pressure in your middle ear. So that tube is really important. If you know someone who's had allergies and quote unquote tubes put in their ears, their tubes put in their ears are in that auditory or eustachian tube to keep the tubes open and from getting overly infected or built up with goo, right? So if you have pretty bad allergies, sometimes you can get mucus built up there, and that mucus can trap pathogens and cause problems. So these eustachian tubes are basically where tubes, quote unquote, are put in the ears when an individual, usually younger kids, have allergies and get lots of infections. It causes problems with hearing because you're unable to regulate pressure in the middle ear. In that cavity, there are also 3 little teeny tiny bones called auditory ossicles. Let's take a look at them and then we'll talk about them. So this section here is the middle ear, from the tympanic membrane all the way to this little window right here. It's called the oval window. And right here is the eustachian tube. So right here would be right like your throat, the pharynx, right? One part of your throat. So that tube, auditory tube or eustachian tube, is always open, but it can get infected. And if that gets infected, sometimes tubes will be put in, but also that little opposite end of the tube at the throat here usually has a little tiny membrane that when it opens ever so slightly, your ears pop when you go up in an airplane. These 3 bones, 1, 2, 3, are the auditory ossicles. And we're going to talk about them individually here shortly. So we've got external ear, middle ear, and then we'll talk about the inner ear, which is where all the sensory receptors are located and encased in the temporal bone. So the sensory organs and sensory cells for hearing and equilibrium, we've talked about them before. They're called hair cells. They receive amplified sound waves from the middle ear. So basically, the external ear collects sound waves and sends those sound waves to the tympanic membrane. And then from the tympanic membrane to the ossicles, those sound waves are then amplified, enhanced, increased. And the amplification of those sound waves is communicated to the inner ear into basically some bony structures called the bony labyrinth. We'll talk about that here shortly and what the heck that is. So here's the structures of the inner ear. We've got this whole section here, which is called the vestibule. We'll talk about here shortly. And then this is the cochlea. So those are the 2 major structures, vestibule and cochlea. And remember, the cranial nerve that innervates this whole inner ear, the vestibule and the cochlea make up the inner ear, is the vestibulocochlear nerve, right? One of the 12 cranial nerves, right? It's cranial nerve 8. And basically the bony labyrinth is basically the bone here that you can see these external structures. Encased in that bony labyrinth is all the really delicate sensory organs. So these ossicles, excuse me, in the middle ear, right here's the tympanic membrane, so the eardrum. Right there's a eustachian tube or auditory tube, so you kind of have an idea of what you're looking at. So this whole cavity is called the tympanic cavity. And these 3 ossicles, the malleus, the incus, and the stapes, you can't see the stapes very well because there's muscle here, and those muscles help control the movement of those ossicles. But basically, these three ossicles are what amplify sound waves coming from the tympanic membrane. So as the tympanic membrane vibrates back and forth as sound wave sound waves hit it, these ossicles move. And there's another little window, membranous window right here, that as these three ossicles oscillate or move, vibrate in here, they vibrate that window. And that oval window, that little thin layer of connective tissue, or of tissue, membranous tissue, is what opens up to the inner ear. And so for communication from the outer ear to the middle ear happens by way of communication from the tympanic membrane to the malleus, one of the three ossicles. The malleus communicates to the incus, and the incus to the stapes. And the stapes, is shaped like a stirrup, communicates to the oval window, which takes that signal and heads into the inner ear. So let's talk about the bony labyrinth. There's actually, within the bony labyrinth is called the membranous labyrinth. So the bony labyrinth surrounds and protects the membranes located in there, the membranous labyrinth. And it's composed of three parts: semicircular canals that are partial circles, vestibule, and the cochlea. So there's bone that surrounds all of these structures called the bony labyrinth. And within those, just deep to that bone, there's a fluid called perilymph. And fluid flows between the bony labyrinth and the membranous labyrinth. So it's a cushion basically in there so that everything is protected. So take a look. So here's the cochlea. Here's the vestibule. And here are the 1, 2, and 3 semicircular canals. So these semicircular canals and the vestibule, we'll talk about later, are involved in equilibrium, and the cochlea is responsible for hearing. But the bone that surrounds it is the bony labyrinth. So there's fluid in here, and then the membranous labyrinth is kind of this blue and purple color. So that's the membranous labyrinth. And within the membranous labyrinth is basically the receptors, the sensory receptors for hearing in the cochlea, and then balance and equilibrium in the vestibule and semicircular canals. So that membranous labyrinth, again, it's basically a collection of fluid-filled tubes and chambers. And the fluid is really important in activating the sensory receptors. So it houses these fluid-filled tubes of the membranous labyrinth, house the receptors for equilibrium and hearing. Remember, hair cells. So those are the big sensory receptors, hair cells. Those fluid-filled chambers are filled with another type of fluid called endolymph. So perilymph is the fluid that's just underneath the bone and outside of the membranous labyrinth. And within the membranous labyrinth is endolymph. And that endolymph, basically the membranous labyrinth is composed of the semicircular ducts within the semicircular canals. Within the vestibule are the utricle and saccule, and within the cochlea is the cochlear duct. So these are the most important structures in hearing and equilibrium, because there's the sensory receptors, the hair cells, are located in semicircular canals, utricle, saccule, and cochlear duct. So really, really, really important. And the cochlear duct spirals like a snail shell. So the utricle and saccule are these two structures here. Utricle comes, basically saccules first, and then utricle closest to the cochlea. The semicircular ducts are in the semicircular canals, right, encased in that bony labyrinth. And here's the cochlear duct, right? Within that duct, there's the major organ of hearing. It's called the spiral organ. That spiral organ spirals along the cochlear duct. So let's talk about hearing and equilibrium first. In the semicircular ducts, encased in the semicircular canals, there are sensory receptors, and there's 3 semicircular ducts that sense changes in response to rotation. Anterior, posterior, and lateral semicircular ducts all respond to rotation in the head. And each duct has an ampulla ampulla is just an expanded or swollen region where there's what's called cristae ampullaris. So that area, that swollen region of the ampulla, there's processes of hair cells in that ampulla that are embedded in something called the cupula. And that's basically a flexible and elastic kind of gelatinous structure. So take a look at the next slide here. So right here's the cupula, this whole kind of gelatin-like structure. And right here are the hair cells. So in this hair cells, when the cupula moves, the hair cells move too. And remember, we've talked about these hair cells. When they move, there's stereocilia right here on these hair cells. And when they bend, they can activate and excite these hair cells to generate action potentials along this sensory nerve. So the movement of this cupula is super important. So basically, here's what happens. The cupula floats in endolymph, the fluid within the membranous labyrinth. And when the head rotates, the movement of the endolymph, the fluid in there, moves the cupula from side to side, and that distorts the receptor processes. I mentioned those are stereocilia, and movement in one direction stimulates the hair, in the other direction it inhibits it. So basically, your head can rotate in any which direction, lateral, anterior, or side to side. Basically, rotation can activate those hairs and tell your brain how you're rotating, turning. And basically, when you stop rotating, the movement stops the endolymph movement and the cupula returns to its normal position. So take a look. So you rotate, basically you rotate your head or rotate your body, and the cupula rotates, or the endolymph moves in one direction and basically distorts the cupula, bending the stereocilia on hair cells. At rest, it's not moved, and then there's movement in the opposite direction that moves it the other way. So basically, the direction of the endolymph, that fluid there, is what moves the cupula, and that movement of the cupula is what activates hair cells. And that's related to balance. Utricle and saccule in the vestibule respond to changes in equilibrium. So there's a structure in the utricle and saccule. Basically, the sensory structure is called a macula. And the utricle, the macula in the utricle is sensitive to changes in horizontal movement, whereas in the saccule, it's sensitive to changes in vertical movement. There's structures within that macula. One, hair cells, the sensory receptors, but the sensory receptors are activated basically or inhibited by way of these crystals called otoliths. So take a look at this next slide. Right here is the saccule and utricle, right? If you blow up, right here's the hair cells. This figure right there is hair cells embedded in those, in the saccule and utricle. Right here are the otoliths. And when these otoliths, so again, this is just a layer of kind of gelatinous fluid. When you bend your head or neck in one direction or another, these otoliths slide and pull the gelatinous structure with them, and the projections on the hair cells bend and can be activated or inhibited. So the mechanism of activation of hair cells is exactly the same, whether we're talking about balance or equilibrium or even hearing, we're going to talk about later. There's just different structures or different types of fluid that are involved in bending those hair cells. So when your head is upright, the otoliths basically sit on top of the membrane and can push hair processes down. But when you tilt your head, the gravity of tilting your head pulls on the otoliths and shifts them to one side. And that movement distorts the hair cell processes and generates action potentials. So take a look. Normally, they just sit on top, right? The hair cell processes don't move much. But you bend your head back, and the otoliths, this purple thing, slides and moves, and along with it, the membrane here moves as well, and it bends the hair cells and basically just pulls them down and activates those hair cells to say, hey, brain, I'm moving my head. We need to reposition ourselves to make sure we don't fall over, right? So there's basically an action potential generated here because these otoliths are sliding down a gravitational pull and bending the stereocilia on hair cells. So let's talk about hearing in the cochlea. The cochlear duct is a long coil that runs through the entire cochlea. So it's basically a spiral shape, and it's got two chambers that coil around the cochlear duct that are filled with perilymph. Remember that protective fluid called the scala. Those two ducts are called the scala vestibuli and the scala tympani. You'll see them labeled also as like the tympanic duct or the vestibular duct. I think your book has it this way. So the bony labyrinth basically encases all three of these ducts, except at the oval window. And the oval window is basically the beginning of the inner ear, where the stapes stamps or connects directly to the oval window and transmits those vibrations, sound vibrations that have been amplified to the inner ear. And that's at the base of the scala vestibuli. So that fluid in there moves around and basically oscillates in the scala vestibuli, because the oval window is membranous. It's not bone. At the very opposite end of the cochlea, there's a round window, and that's at the base of the scala tympani. And the round window is another layer of connective tissue that is able to kind of bend and flex, so that as the fluid increases or decreases pressure in the cochlea, it can respond. And then in the cochlear duct, there's hair cells, the sensory receptors, embedded in an organ called the spiral organ. Remember the cochlear duct spirals? It's called the spiral organ or organ of Corti, and it runs the entire length of the cochlea. So take a look. So here, up here, there's the vestibule, semicircular canals. Right there is the stapes, one of the three ossicles, right? Auditory ossicles, those three bones. So as the stapes vibrates back and forth, it pushes in on the oval window and pushes the fluid in the scala vestibuli in this circular motion, right? So remember, these are all connected, and this blue is the cochlear duct in the middle. So the purple is the scala vestibuli. Fluid moves all the way up here, gets pushed around, and then the scala tympani runs in the opposite direction, and as it hits the round window, this will kind of bulge out. So as the stapes is pushing in on the oval window, that fluid in the opposite direction is pushing out on the round window, and they can basically oscillate back and forth with one another to maintain pressure within the cochlea. If you cross-section the cochlea, where this green plane is, and look inside, right here is a scala vestibuli. Right here is a scala tympani, and right here's the cochlear duct. This little structure here is the organ of Corti, or spiral organ, and I think it's pointed out over here, right? So here's the three ducts pointed out. The organ of Corti, to help you remember which of these ducts is which in this cross section, the organ of Corti kind of points up always towards the scala vestibuli, and there's a membrane right here called the vestibular membrane that separates the scala vestibuli from the cochlear duct. So we'll talk about that on the next couple slides. But I want you to see this first, because this is an actual histological slide of the cochlea. Look how much different it looks. So right here is an organ of Corti. And there's an organ of Corti. And there's an organ of Corti. This is the vestibular membrane. So this is the scala vestibuli and the scala tympani. Looks completely different, right? Or not completely different, but very different. So this spiral organ or organ of Corti is so important. It is literally your organ or your sensory organ for hearing. And it is separated, the cochlear duct basically is separated again from the scala vestibuli by the vestibular membrane. And on the opposite side, the basilar membrane separates the cochlear duct from the scala tympani. So there's two membranes that actually just separate these ducts. But remember, the spiral organ and the cochlear duct they are part of and basically embedded in the membranous labyrinth, which makes up the cochlear duct itself. There is another membrane, a third membrane called the tectorial membrane that rests on top of the spiral organ. It's part of the spiral organ, but it rests right on top of the hair cells. Remember, the hair cells have little tiny projections called stereocilia, and they need to move to be activated or inhibited. Well, this tectorial membrane moves and pulls or pushes on the hair cell projections, and that's what stimulates them to generate action potentials. And then remember, the vestibulocochlear nerve is that cranial nerve that these hair cells send action potentials, basically communicate to send action potentials back to the brain. So this spiral organ, take a look at it. So here's the scala vestibuli. There's the vestibular membrane again that separates the scala vestibuli from the cochlear duct. There's the spiral organ and the basilar membrane that separates the cochlear duct from the scala tympani. So if you blow that up, the smaller membrane, remember the tectorial membrane that we talked about on the last slide, covers the hair cells. And these hair cells, again, are your sensory receptors. These stereocilia, as fluid moves in the cochlear duct, the tectorial membrane moves and shifts and bends these stereocilia. And as they bend, the mechanoreceptors, basically located there, activate, generate action potentials, and send an action potential signal along the sensory neurons. This would be the cochlear branch of the vestibulocochlear nerve. And this slide just summarizes what I've been saying. The sound waves at the tympanic membrane trigger pressure changes in the paralymph in the cochlea. So at the tympanic membrane, which is on this slide, the tectorial membrane, right? So the tectorial membrane, basically, sound waves at the tympanic membrane trigger pressure changes in the paralymph in the cochlea. And the basilar membrane bounces up and down in response to those pressure changes. So hair cells are pushed up against the tectorial membrane and distorted. So take a look here. This is a spiral organ. So pressure waves in the paralymph push up on the basilar membrane, and that pushes up on the hair cells and forces the hair cells into the tectorial membrane, and they bend. And that bending is what allows them to generate action potentials, because there's mechanoreceptors on those cells. Those action potentials are sent back to the brain to communicate about hearing. And there's basically a step-by-step process on this slide here that I will let you kind of go through and understand. But basically, from start to finish, sound waves make their way in, hit the tympanic membrane. Those sound waves, the tympanic membrane vibrates, and that then vibrates and moves the auditory ossicles. As the ossicles vibrate and amplify the sound waves here, they are then communicated to the oval window, which opens up to the vestibular duct, or the scala vestibuli, right? So movement there opens up to the scala vestibuli. That paralymph in this fluid here spirals up and then into, this is now the scala tympani, and pushes down this way out towards the round window, right? Pressure waves, as this fluid moves, basically distorts, remember, the basilar membrane in the cochlear duct, this blue in here. And as that membrane gets distorted, the processes, the stereocilia on hair cells, bend and generate action potentials in the hair cells. And that information is sent along the vestibulocochlear nerve back to the brain. So it's all summarized right there. Before we leave, talk a little bit about some disorders like vertigo. It's basically an illusion of movement. So your body's not moving, but something in your inner ear is telling you that you are moving. It can be pretty complex, actually. The internal ear receptors, because of think about the complexity we just talked about, those hair cells, and they have to bend in response to different movements of different structures. Anything that disturbs the endolymph, even like flushing the external acoustic meatus with water, or excessive consumption of alcohol and certain drugs, basically the vestibular branch of the vestibulocochlear nerve, it can be involved, and sensory nuclei in pathways in the central nervous system can be involved as well. So basically anything that disturbs the fluid movement in those ducts can cause an illusion of movement, even though you're not. With relation to hearing disorders, there's two different types of deafness. Conductive deafness, which basically means there's some interference with the transfer of vibrations from the tympanic membrane, which is your eardrum, to the oval window. So the nerve is fine, the inner ear is fine, typically healthy, but there's something blocking the external ear or the middle ear from getting sound waves, transmitting sound waves to the inner ear. That could be caused by even excess wax buildup, trapped water, scarring of the tympanic membrane. So if you've ever had a burst eardrum, as it heals, it can scar and it won't vibrate as easily or as efficiently, or immobilization of the auditory ossicles, so that malleus, incus, and stapes won't vibrate for some reason. So that's conductive deafness. Nerve deafness is a problem with the cochlea, or something related to the vestibulocochlear nerve, so along the auditory pathway. And that can be caused by really loud noises damaging the stereocilia on the hair cells, or a bacterial viral infection. And hearing loss, I guess everything gets worse as you get older, right, guys? And this occurs with age, accumulating damage from loud noises. So think about like listening to loud music in your car for day after day after day and year after year. But also the tympanic membrane, the eardrum, gets less flexible over time. And then stiffness in the auditory ossicles also occurs. And also ossification. So basically the round window starts to become more bone-like, and that can prevent the movement of fluid in the inner ear, which basically prevents hearing. All right, I will see you in when we talk about the eye. See you soon, guys.